Okay. Yeah. Hi, thank you for joining. So I will start with today's session, and I hope my screen is visible to you. You can let me know in the chat box if it's not. Right. So welcome to the fifth problem solving session for the course Cellular Biology offered by NPTEL. And like the previous four problem solving sessions, this also will include questions on previous year assignment and some exam pick questions so that it can help you understand these topics better. So let me begin. So week 5 was so all so far we have been discussing about mitosis and about how DNA replication test takes place, about the cell cycle as a whole. So we'll move one step ahead in cell cycle this week, and we will be studying about apoptosis and meiosis, and that topic is further divided into 4 lectures if you remember from the lecture notes. The first lecture for this week had cell death, aging, and senescence. The second lecture was about apoptosis in general, and then there were 2 lectures which are for meiosis, right? So we will be covering more about meiosis, but before meiosis, we will be looking into the cell death pattern. So how does cell death takes place? We know that there is apoptosis and necrosis, and then we'll also look into cell aging and senescence. What is a senescence actually, and what is the difference between senescence and apoptosis? And we'll do apoptosis a bit more in details, right? Okay, I'll begin the question explanation for the lectures because we then will be on time. So the first thing that we will discuss, I will go as per the lecture series. So first will be cell death, aging, and senescence. So let's begin with that. So we already have an idea about what is apoptosis. We know what's necrosis. We did discuss apoptosis previously, right? Now, but we will do this in a bit more detail in this session, right? So let's begin. So cell death basically, as the name suggests, it basically leads to the death of the cell. And then people also can think of it as a cellular failure, that it has not functioned properly, so it has led to its death. But that's not exactly a way that can properly capture the breadth of the topic, because just saying cellular failure would seem like anything that is not functioning, and anything which is randomly dying, which we also discussed when I was discussing about apoptosis, right? So the proper way to define cell death is basically to say this as a regulated biological process essential for development, tissue homeostasis, and disease prevention. The key word here is it's a regulated biological process. You have several checkpoints, you have several regulation points which you are uh, which the cell detects for the activity. And if it is not matching the qualities, when you go ahead and there's a cell death, there's a it's also called, if you remember, apoptosis is also called programmed cell death. The programmed is because of this reason. The first, it is detected, and then there is a pattern of or a series of reactions, uh, pathways which takes place to ultimately uh, kill the cell, right? So apoptosis is one of the way or one of the type of cell death, which will be anyway doing in the next part. The other type is necrosis. So there are 2 ways of a cell death. One is apoptosis, which is also called programmed cell death. The other is what we also discussed, necrosis. And necrosis is basically, if you want to see necrosis, you will see a patch of skin which is blackened out, which the cells have got really dead, the skin there is dead. So that's necrosis. And why are those looking like this? They're looking like this because the membrane of the cells there have lost their integrity. There is cell swelling there. So necrosis is characterized by cell swelling. So there's local swelling. Let's say if there's an injury there, so you will see a blackened patch. You will see local swelling. You will see organelle rupture, inflammation. That's a key response or mechanism in necrosis. So in necrosis, there is the difference between necrosis will and the apoptosis will be that apoptosis is basically triggered by intrinsic or extrinsic factor both, and the mechanism of necrosis and the mechanism of apoptosis is very different. One includes ATP depletion, one includes cell swelling, while the other, which is basically apoptosis, it includes DNA fragmentation as a cue to then release of cytochrome C from mitochondria, which we will discuss in the next part anyway, right? So you have 2 types of cell death, which is basically your apoptosis and your necrosis. So this partly covers what we were talking about, cell death. And now moving to aging. when do you call a cell is aging? When you say that cell is aging, aging is something which is marking the cell for, or it's telling the tenure of the cell. So it's basically telling that this cell is about to die, or this has some problem and it needs to be fixed, right? So if you remember from your lecture notes, there are 9 hallmarks of aging. A cell is said to be aging if it shows these 9 hallmarks. The 9 hallmarks, if you remember, they were genomic instability. So you have some kind of problems in the genome, and your genome basically tells you that it is not proper. There is some misalignment, there is some gap in the genomics, in the genomic sequence, or there is some length shortening, or there is some intransversion, inversion. So anything which is to do with genomic instability marks it as cell being aged. Then you have telomere attrition. Attrition means shortening of telomeres. So you have telomeres which if undergo shortening in their length is basically a sign of aging. Then we had also seen about epigenetic alterations. So if you remember about methylations, DNA has methylation as epigenetic modifications. So if there is any change in the pattern of those epigenetics, if you basically if you read about epigenetics, if the particular gene has methylated conditions in its normal functioning, when it is in the new cell also the same epigenetic pattern is imprinted. So if it is methylated there, it is methylated in the next or the daughter cell as well. But that when it doesn't happen, when your genomic imprinting is not proper, there is epigenetic alteration, that is also a mark of aging, right? Then you have loss of proteostasis. Is basically the the basically the content of the proteins, the cellular homeostasis is lost. So that is also, of course, a stability, a sign of aging, because the cell is not functioning properly. If you remember, Professor had discussed about nutrient sensing. So if a cell is experiencing deregulated nutrient sensing because of a protein like MTAOR or AMPK, that basically it's not able to uptake appropriate amount of nutrient, for example, glucose from its vicinity, it's a sign of cell aging. We have mitochondrial dysfunction when your mitochondria doesn't function properly, and that makes perfect sense because mitochondria is basically the ultimate site for energy production, ATP synthesis. And if your mitochondria is not functioning properly, and for many other reasons, that's one of the reason is ATP production, but there are so many other functions. It has so many protein complexes that are part of the important pathways of cellular metabolism. So if it doesn't function properly, your cellular metabolism gets affected and your cell also doesn't function properly, right? So that's also a sign of aging. Cellular senescence, which we will do in the next topic, so that is also a sign of aging. Then you have stem cell exhaustion. So if you remember, stem cells are bank. Stem cells are basically those cells that can like propagate or proliferate, differentiate into different kind of cells. So they are basically the stock of cells which replicate whenever there is demand. But if your stem cells are exhausted and you're not able to fill them again, or if they're not getting uh, like uh, so they are being used up but they're not being deposited back, then you have less of stem cells, you're exhausting your stem cells bank, and that is also a sign of aging because you cannot regenerate anymore. And the last one being altered intracellular communication. So if your cell to cell communication, which is a very important field or very important process for your maintenance of cellular environment, for example, cells interact by so many chemical components like chemokines, interkines, and then you have cell to cell junction, which holds cell up together. So all of these, they have something called contact disjunctions. So if you have cells contact each other, they stop growing. These are all part of normal cellular metabolism, cellular biology. But if all this communication also altered, that it's also a sign of cellular aging, which basically means that the cell is not able to function the way it is, right? And all these basically come from their problems associated within the cell. For example, there can be DNA damage accumulation. So, for example, you have replication error, which we also discussed while discussing DNA replication. So, you have replication error, you have oxidative stress, you have inefficient repair mechanism, which is something that we spoke about checkpoints when we were discussing checkpoints, that if there is DNA damage, so a cell otherwise tries to correct the all the anomalies that is observed. So if it's replication related, it will stop the replication, it will repair the DNA, it will repair those anomalies that it has observed. If it is not about DNA repair, if it is about something else, if any other stage, it will try. So all the hallmarks are basically when the cells have are not able to correct it anymore on their own. And that's the term literally what it's trying to show, right? When we say that humans have aged, we are basically saying that they are not able to do the function that they were doing so far in their life properly. You are when you are aged, you are not able to do simple jobs that were very easy for you otherwise. Similarly, for cells also, like I said, cells will always try to repair any anomaly that it sees. But all these 9 hallmarks are basically they show up when the cell cannot correct those anomalies on their own, and the cell has quoted aged and it cannot function properly anymore. So that's the idea there, right? And what happens because of this aging when the cell is not able to do all of these functions that it's supposed to do anymore? There will be reduced proliferation, no stem cells. That's why there will be metabolic shift because your cells are not uptaking glucose as much or nutrient as much as it is supposed to. So there will be always a depletion in the energy requirement. So there will be a metabolic shift. There will be inflammatory signaling because your cells are not functioning properly. They will be under stressed conditions. So there will be inflammatory signals. They will be in, there will be because there will be reduced proliferation. There also will be reduced regeneration, and as I said, there will be stem cell depletion. So all these are basically guiding the cell towards death. They are basically saying that the cells are not functioning anymore, and in the coming time they will be dying very soon, right? And the last thing that I said, cellular senescence, which is also a marking for aging, is basically a permanent cell cycle arrest, which is triggered by stress. So if you remember in the cell cycle, there was this offshoot which was showing that it goes to senescence, which is basically though not quiescence. Quiescence can still come back into cell cycle. The ones which are permanently undergoing cell cycle arrest, those are called senescence. And when does the cell decide that okay, henceforth I will not be part of cell cycle, I will not be undergoing DNA replication or any division for that matter? The ultimate cues for senescence are telomere shortening or oncogene activation. If you know oncogene, these are cancer genes. So they have basically oncogene and activation puts the cells under permanent cell cycle arrest. There can be DNA damage and oxidative stress. Now you will say that these terms are all intertwined with each other. Yes, because nothing is working alone, nothing is working on its own. Everything is affecting the other. If there is DNA damage, if there is stress, if there is stress, then there is other metabolism effects. So all of these are linked to each other. That's why you cannot tease them out. But all the 9 markers there which we just mentioned, those are not independent in their functioning. They are basically, just a second. Yeah, so they are basically all together what is making up the hallmarks of the cell aging, right? And when we will be discussing in the slides also as the questions that there are 2 pathways which are responsible for senescence. One is P53 and P21 pathway, and the other is P16 and RB pathway. So in the first one, P53, you have DNA damage. We did this bits and pieces in previous sessions also. You have DNA damage that basically activates P53, that further activates P21, and that goes and does inhibition of CDKs. What are CDKs? They're important for cell cycle progression. So when you see DNA damage and P21 goes as inhibits CDK, your cell cycle arrests in the G1 stage, right? And then you have the replication, the DNA repair mechanism into play. The moment the damage is reversed, the cell goes into cell cycle again, right? And the other one is also for cell cycle, which is P16 and RB, retinoblastoma pathway, which is again acting by inhibiting CDK, which is in this case, it is the CDK4 and 6, right? Now, there are few terms. So you will see some terms on the slide also, which are also called SASP, which also you must have encountered in your lectures, which are called the senescence associated secretory phenotype. So these are basically those cells which are basically showing the hallmarks of aging, and they are then entering into this stage of permanent arrest, cell cycle arrest. So they will secrete some proteins, for example, or some chemical compounds, for example, interleukin 6, interleukin 8, for example, I'll show you here. You have some cytokines, chemokines, growth factors, proteases, ECMs, right? So they have growth factors, all of this. Now, what are these trying to tell? These are basically giving signals to the nearby cells saying that this particular cell is undergoing chronic inflammation. Chronic inflammation is basically prolonged inflammation, and as we told that in senescence, inflammation is one of the signals, right? We also, these signals show that there is tumor progression, which is basically oncogene activation, right? This also, because of growth factors and because of ECMs, it also implicates tissue remodeling, which is basically your senescent cells basically look like this, right? So they are these bleb shrink cells. So this shrinkage happens because of all these factors that take away the cell's membrane integrity, right? So your cell, basically the one which is senescent, is basically shrinking. So it's also called tissue remodeling. And then you have all of these other factors which also come into play for your communication, right? So now on this slide, for example, you can see that there are a lot of factors which are written with plus and minus. So what does it say? Your senescent cells basically has these two type of effects, right? So the plus ones which are here, these effects are ones which are also in blue in this case here. So what these are basically the beneficial effect which can come with the early stage of senescence. So when a cell is undergoing senescence, some of the so these chemicals that they secrete can have some beneficial effect. For example, they can have tumor suppression. So they can, these senescence basically prevents them from proliferating the damage and oncogene activated cells. So they have oncogene activated, so they undergo the cell cycle arrest, so they don't proliferate anymore. So they can act as a barrier to cancer, right? Then they have wound repair because of the tissue remodeling and immune cells, immune activation, all of this. But when the senescence has prolonged, or then there has been a chronic senescence, that's when they start becoming harmful, and that's when the same action which was being beneficial for the cell now starts behaving as a harmful effect. For example, now after a certain stage, it starts promoting tumor. It starts to like implicate tissue dysfunction, immune evasion, stem cell depletion, and then it is basically now that the cell has truly aged, and now it will be marked for death, right? So that's all about it. And also this one is something that you would have seen. This one is basically telling the different kind of events from different kind places of the cell. For example, you have nuclear events, and you also have these cytoplasmic changes. For example, let me show you. So these are nuclear changes, which basically includes DNA damage, telomere shortening. Telomere shortening is basically here. Telomeres are at the end of DNA, so your telomeres are shortening from your ends. Then in cytoplasm, you might have ROS. ROS basically when you have these chemical interactions in the cells, these oxidative reactive oxygen species comes up, and these are very, very like they react very quickly, so they can damage the cellular organisms very quickly. So that's why ROS is a problem. And then you also have mitochondrial dysfunction. So all of these, along with chromatin remodeling because of associated problems like heterochromatic foci or cytoplasmic chromatin fragments. So all of these different kind of names that you are seeing here in bold, all of these together basically go up and mark the cell for senescence. They are signs, they are the true uh, phenotype of senescent cells, right? And then when they are in the senescent stage, they undergo cell cycle arrest. Initially, it's fine, but as they are prolonged, they will lead the cell to death. That we will do in the next set. But let me first try to, like get these at the same place. Okay, so let's try to answer some questions so that we can make some sense out of these questions. Okay, right. So there are 4 questions on this slide. The first one says, which of the following best explains why genomic instability? So like I said, there are hallmarks, and we can discuss this as the question progresses. You will understand these hallmarks better. The first one is, why do you think genomic instability contributes to aging? Telomere attrition, which is basically shortening, is why the shortening happens. What is basically a Hayflick limit? And replicative senescence is triggered by what? Directly. Most directly means all of, I just told you everything works in concert, but which one directly affects the cellular senescence? Please use the chat box for your answers. We will stop at this slide for 2 minutes and then we'll proceed ahead. You can use the chat box. So I have one response, CBCC. Anyone else? So all of these for now on the slides are basically signs of hallmarks of aging. So why is each of them a hallmark of aging, or why are each of them even taking place in the cell? So maybe a minute more and then I can give the answers and why they are like the correct answer and the discussions. Okay, I will just move ahead. Right. So C, B, C, C. Okay, so let me look at the answers. Right. So let me just explain them. The first one says, which are the following best explain why genomic instability contributes to aging? So why should you basically, why should any problem with the genome contribute to cell being aged, right? And that's because the C option, which is saying that it is accumulating, it causes accumulation of DNA damage over time, right? So any genomic instability, genome means something to do with DNA, and anything which is basically related to genomic instability is basically leading to accumulation of mutations and DNA damage. And as these are problematic for cellular function, these contribute to aging, right? Okay, telomere attrition primarily results. Why is there shortening of telomere? It's because the DNA polymerase is unable to fully replicate chromosome ends. So when we did DNA replication, you would remember the telomere, the DNA polymerase moves ahead, copying the new, adding the new bases based on complementarity. But the function of the processivity of the DNA polymerase is reduces when it reaches the end. That's why at the end you always have the end portion of DNA is called telomere, and it always, you will see that because of this end replication problem, there is a consistent shortening of telomere over the cycles of DNA. Right. So it is basically referring to that a particular cell can only divide up to a certain time. Okay, and that's mostly 40 to 60. Why? Because after that, it is unable to properly replicate and use up the nutrients around it, around its surrounding, right? So when the cell does not do that, that's when a cell is problematic, right? And lastly, replicative senescence is mostly directly, so the name suggests replicative, and it's basically critically short telomeres. The senescence basically is where the cell cycle is arrested. But here, when we are defining senescence, we are also giving it a name. We are calling it replicative senescence. That's why it is directly triggered by short telomere. Right. Next set. So telomere dysfunction induced foci. Okay. What are these referring to? Loss of proteostasis, which is again a hallmark of aging, primarily affects what? Deregulated nutrient sensing in aging is strongly associated with alteration in which pathway, which I mentioned briefly while passing, and mitochondrial dysfunction contributes. Basically, all these questions I have put here collecting over the past assignments and new questions and exam questions for you to understand the cell aging and the hallmarks of cell aging better. So we don't just name the hallmarks, we also understand why are they actually being called as a hallmark. So even if you don't know any answer particularly or you're doubtful, it's all right if you write it. If you don't write it, the idea is that you try to solve it and try to remember as much as from the lecture notes, because these are the kind of question that cell biology, especially this course, will focus on. I told you last time also, you should remember the components and the constituents of a pathway, which protein is involved in with what kind of functionality. That is very important. Anybody wants to answer? Okay, anyone else? Or I hope you guys are at least doing it at your end, even if you are not typing out the answer here. Okay, I will wait for 30 more seconds and then I will just show the answers. And you can match if you were thinking in the correct directions. Okay, so let me give the answers. Okay, so the first answer or the first question here is telomere dysfunction induced. So anything which is to be done with telomere problems. So telomere dysfunction induced foci, these are basically characterized by DNA damages, because anything has to be that is to be done with telomere has to be done with DNA. So DNA damage response proteins localized at telomeres. So any problem with telomeres, foci is basically the uh, patch or the location where the problems are induced, and which is basically triggering the response for the cell metabolism here, right? So here we are talking about aging. So we are talking about telomere dysfunction induced foci, and these are basically enriched with proteins which are responding to DNA damage. So when you have DNA damaged proteins, the cell basically enters senescence, right? And what are these proteins? These are basically something called gamma H2AX, which are basically associated with the centromeres or the histone proteins. Centromeres, the histone proteins, and a different kind of proteins which are found around the telomeres, right? Okay, next one says loss of proteostasis during aging is basically affecting the protein folding, degradation, quality control. I told you of the homeostasis of the cell, which is to be doing which is to be anything to do with the proteins in the cell, be it their synthesis, folding, or degradation. Anything that is problematic to the protein functioning is basically lots of proteostasis. Stasis means balance, and proteome means protein. So proteins functioning balance. Okay, you can remember it like that. Next one, deregulated nutrient sensing in aging is strongly associated with alteration in which pathway? I had mentioned MTOR pathway. MTOR pathway is basically uh, important pathway for the cell growth, proliferation, and survival, even protein synthesis. Okay, so that pathway is affected the most during deregulated nutrient sensing. And the last one here says mitochondrial dysfunction contributes to aging via elevated ROS production. ROS is basically, as I said, reactive oxygen species, which are not good for cell health. And then you also have, along with these, you have DNA damage, protein lipid, because you can imagine if mitochondria is not functioning properly, you will also have problem related to the DNA, related to protein and any other biomolecules in the cell. Okay. All right. Next part. So now moving further ahead into senescence and aging, defining feature of cellular senescence. When you say cell has undergone senescence, what is the first thing that you will, or what is the most important thing? P53 I mentioned. So activation of P53 during senescence leads to which of these following options? The SASP involves which of these compounds, and a stem cell exhaustion. So again, you can see there are multiple questions about hallmarks, and I hope that helps you when you are preparing for exam, because these are basically helping you revise the topic again. So for example, the second question we've already discussed, so this shouldn't be that difficult, or at least not very unknown, or something that we haven't like discussed yet. Any answers? I have one response, which is CBC. Anyone else here? Okay, I will go to the answers since we need to move to other topics also. Right. So when you are defining senescence, the very important thing is permanent cell cycle arrest despite mitogenic signals. What does it mean? That the cell will have lot of cues for proliferation. Mitogenic is basically factors which promote mitosis. So even though there are many factors in the vicinity which promotes mitosis, the cell will not be able to undergo cell cycle, or it will not go under cell cycle. It will undergo cell cycle arrest. And that's when you know that even the cues are not functioning. The cell has truly undergone senescence. Okay. Activation of P53, this is something which I just discussed, P53 pathway and P16 pathway. So the P53 pathway works by activating P21, which inhibits CDK and enforces cell cycle arrest, right? The SASP involves increased secretion of inflammatory cytokines and proteases. Why the other options are incorrect? The first one said reduced cytokine secretion, which is incorrect. It is actually enhancing the secretions. Increased DNA methylation. It has got nothing to do with epigenetic alteration effects at all. And telomere stabilization, that is not senescence. The senescence, one of the keys of senescence is that it is undergoing telomere attrition, right? So that's why the answer will be here that SASP basically involves pro-inflammatory cytokines, chemokines, growth factors, and proteases, which will all affect the tissue microenvironment and then do all the effects like tissue remodeling that I had mentioned about, right? Okay, and the next question was, last one here was, stem cell exhaustion contributes to aging. So when I said that there was no stem cell, it's basically meant that there was reduced regenerative capacity of tissues because there is a decline in the stem cell number and the stem cell function. There is no regeneration, there is no repletion. There is no refilling. It's getting depleted. The stock is getting depleted, right? All right, so let's move to the next part. Right, these are set of questions from I think previous year's assignment. Okay, so this one says, which of the following is not a hallmark of aging? Which of the following statement is incorrect about apoptosis? Senescence is accompanied by which of these options? And caspases, which is something that we will discuss in the next part, is basically what kind of proteases. Caspases is basically from apoptosis that we will now be discussing. Anyone wants to take a attempt? Okay, people are still typing and thinking. I'll wait for some time. CACC. Okay, anyone else? Okay, let me give the answers. So C, A, C, and C. That's correct. Which of these is not a hallmark of aging? Telomere lengthening. That's because telomere shortening or attrition is the hallmark of aging. Rest everything is correct. Okay. Which of these is not true about apoptosis? Apoptosis of cell induces inflammatory reaction. Remember when I was talking about apoptosis and necrosis, this was the very point that I said differentiated between apoptosis and necrosis. Inflammatory reaction is from necrosis. Okay, senescence is accompanied by cell cycle arrest. Okay, decreased ROS, definitely not. Increased apoptosis, the cell doesn't go apoptosis yet. Senescence is basically cell cycle arrest. Okay, and inactivation of CDK inhibitors. No, we want CDK inhibitors. So we have this is basically this takes place because of the inhibition takes place because of senescence. So there is no inactivation. So the true correct answer is cell cycle arrest. And caspases that we will see now is basically class of cysteine protease. Okay. Okay. Now the last two questions here to connect you back to apoptosis and this topic. Which of the following is true about cytochrome C released from mitochondria? We'll see where it is happening from. And which of the statement is true? I will wait for some time for your answers. Okay, anyone else wants to answer? People are typing D and B. Okay. I will wait 30 seconds more and then we can discuss the answers. Okay. All right, I'll give the answers here. Okay, so the first one is which of the following is true about cytochrome C release from mitochondria? We will see in the upcoming slide. It triggers caspase activation. Which caspase? And so we will see there are two different pathways for apoptosis and which one is activated by this. And second one, which of the following statement is true? Death induced by FAS or TNF, which we will again associated with one of the pathways of apoptosis. It causes the activation of caspase 8 via death receptor complex. So we will see all of these now. There was just an initiator to what exactly we're going to discuss now. Right, so now we are going to discuss about apoptosis, which is basically also called the programmed cell death, right? And as you can see on this slide, this is basically showing you the different ways, pathways here, how apoptosis takes place and what happens in a cell. So let me just first read you through this and then I'll explain in detail. So as you can see in apoptosis, you have a normal cell and the cell then begins to shrink. Okay, your chromatin condenses. So the cell is being marked for death, right? So the first thing that happens is cells start shrinking, which is something we also saw in the previous part. Okay, then membrane starts blebbing, is basically making these protrusions. Basically it's basically the entire compact cell mass is now degenerating and it's just shrinking it as a mass, right? Organelles also start to disintegrate. So here your DNA started getting condensed. Here your organelles are getting disintegrated. Next stage, you have your nucleus and organelles collapsing, and the membrane continues to bleb. Membrane is again forming this protrusion kind of. Look at the cell here, and look at the cell here. There, the cell is gone. Okay, and then you have these apoptotic bodies, which are ultimately eaten up by macrophages. So they are marked for phagosome complexes. So they are basically ingested by the macrophages. And this is why, because there is no signals or there is no cytokines which are released from the apoptotic bodies, is why you don't have inflammation, which is what happens in necrosis. In necrosis, the damaged cells release cytokines and chemokines and interleukins, which leads to this cell swelling. Okay. All right. And here are the different pathways in which I will explain to you now. Okay. So let me just begin with first explaining again, once again, to people who've joined later. Apoptosis is basically defined as a highly regulated, energy dependent form of programmed cell death. It's not a random process. It's very much regulated and designated steps are there in that entire mechanism. And this is essential for development, tissue homeostasis, and elimination of damaged cells. We are right now dealing with damaged cells concept, but it is also equally important in tissue homeostasis and development. A simple example, your digits, your fingers are all separated from each other. And that's because these parts have undergone apoptosis. These are disjointed because of this very region. Okay, so that's why you need the apoptosis even during development. That's just how one of the, like reasons. Then in tissue homeostasis, you have this bone structure forming, you have this tissue remodeling happening. So you need some cells at one place and not the others, or you need one cell of layer of cell being replaced by others. So you need to mark those cells for damage. So that is also important. That's also taken place because of apoptosis. Okay, but right now what we will be dealing is with the damaged cells version, which is what is in the line of this topic, but the same mechanism applies to all of these different conditions. Okay, now as I mentioned, apoptosis has two pathways: intrinsic pathway and extrinsic. As the name suggests, intrinsic is something from within the cell, and extrinsic is something which is from outside the cell. Now, something which is inside the cell is basically governed by, as you can see here, intrinsic pathway, basically by this mitochondria. So it's also called mitochondrial pathway. And what is basically triggering this internal pathway or intrinsic pathway? Anything which is to be done in the internal components. For example, DNA damage, oxidative stress, growth factor withdrawal, or ER stress is basically endoplasmic. So anything which is got to be done with the organelles or inside the cell, and it's not functioning properly, that triggers apoptosis, that is intrinsic pathway, right? Extrinsic is something which is coming from the extracellular matrix or outside the cell environment. So you have some receptors which basically bind to ligands, which basically target the cell for apoptosis. And because there is a receptor involved, which binds to a particular ligand, which marks it for death, this pathway is called death receptor pathway. You can just simply remember it like this. Involves a receptor has to be here, and a molecule binds to it. For example, here you can see it's called TRE, or it's also called FASL, or it can also be tubule necrosis factor, TNF alpha, right? So all these come and bind to the receptor and then mark it for death. So it's called this death receptor pathway. Okay. So basically, the way this pathway are activated is different, but ultimately you will see that they both work by caspases, different caspases, but the mode of action is activating the caspase enzymes. Okay, so let's do them one by one. Let's do first intrinsic pathway, which is because of your mitochondria or because of your DNA damage. Okay, so let's begin the first with DNA damage. So you have DNA damage, you have activation of P53. Now P53 upregulates pro-apoptotic proteins and downregulates anti-apoptotic proteins of BCL2 and pro-apoptotic proteins of Bax and BAL, which you see here. Okay, so you have intrinsic DNA damage, ER stress, and hypoxia. They basically go and work on these different kind of proteins, upregulate apoptosis promoting protein, which are Bax and BAC, and then downregulate the proteins which are anti-apoptosis, for example, BCL2. Okay, remember, lot of names will come your way. Try to keep them in mind, because that's what is integral to this part of your syllabus. Okay, so especially remember your BCL2 proteins, pro-apoptotic, anti-apoptotic proteins, which ones are pro, which ones are anti, and what do they do. Okay, right. The first thing is DNA damage, and then you have more of pro-apoptotic. And pro-apoptotic means proteins which help in apoptosis, or which promote pro-apoptosis. So promote apoptosis. The other ones, anti-apoptosis, are ones which inhibit apoptosis. So DNA damage, activation of P53, and these two processes. Right. Next is your mitochondria. So your mitochondria, basically, how does the mitochondrial problem starts? It basically, the membrane of the mitochondria become permeabilized. Basically, they become more permeable. They become this. So this layer here starts becoming permeable. So they become more. So otherwise, it is very selectively permeable, right? So now it is becoming permeable. It's becoming whole. It has, the structure has disintegrated. So this is called MOMP, mitochondrial outer membrane permeabilization. And here, the pore formation in these membranes are because of pro-apoptotic BCL2 members, which are Bax and BAC. So the P53 says, hey, you get activated and go first by starting damaging the mitochondria itself. Okay, so the mitochondria has pore. As soon as mitochondria has this pore on its membrane, cytochrome C releases. Okay, that's the question that we answered before, that mitochondria senses damage and it releases cytochrome C along with other proteins like SMAC and Diablo. Okay. Now, once cytochrome C is released, which is what you see here, okay, the purple ones here. Once it is released, it forms apoptosome. Anything I said last time also, some means the complex taking for that process. So apoptosis complex is apoptosome. So you have the cytochrome C. Now it binds to APAF1, which is a protein, and it requires ATP. That's why it's a regulated energy required process. And once it does that, the once this apoptosome is formed, it basically activates caspase 9. So remember, intrinsic is caspase 9. Okay. But caspase 9 is not the ultimate activator or ultimate function, uh, carries out the function of apoptosis. Caspase 9 is an initiator caspase. It further goes to executioner caspase. There are two caspases, one which is initiated by the pathway and one which takes all does the activity. So the one that kills the cell is called the executioner caspase. It is the caspase 3. So 9 and 3. Okay. And now we'll quickly do the extrinsic pathway, which is this side on your figure. So in extrinsic pathway, there is, as I said, it is a death receptor pathway. So you have these fast L or TNF, they bind to their respective receptors. As soon as they do that, they form something called death inducing signaling complex. Okay, DISC. So you have the receptor, you have the protein, and then you have procaspase 8 coming into play. Procaspase 8 goes to activate caspase 8. Now, what you need to remember here is caspase 8. Okay, this directly activates caspase 3, which is basically the executioner caspase. So one pathway, caspase 9, caspase 3, the other pathway, caspase 8, caspase 3. This is what you need to remember here, the names clearly. Okay, now what does executioner do? What does it do? It basically leads to DNA fragmentation, chromatin condensation, nuclear fragmentation, membrane blebbing, formation of apoptotic, all the processes that I showed you here are being taken up because of these and this gas spaces. Okay, so they are doing these activities. Okay, other than that, few other proteins that you need to remember in apoptosis, as I said, anti-apoptotic proteins, pro-apoptotic proteins. You also need to remember inhibitors of apoptosis proteins. You would have come across these terms, IAPs, inhibitors of the apoptosis proteins. So these are all for regulation. Okay, and then you have caspases 9 and 8. And executioner 3 is what I discussed. You also have 6 and 7 as executioner. Okay. Now, what is the importance of apoptosis? If you have too little apoptosis, too little death of cells, you will have cancer or will have autoimmune diseases because cells are not dying. But then if you have too much of apoptosis, you have too much of cell death, it is causing neurodegeneration. So that's why apoptosis is regulated and it is under balance always. Okay. So, and last thing, what is basically these anti-apoptotic and pro-apoptotic? As I already mentioned, these are basically your BCL2 family proteins, and one is promoting apoptosis, one is anti-apoptosis. Okay, so you need to be very clear which ones are which and which ones are activated or or repressed by the P53. Okay, all right. So let's try to answer questions. It will definitely become more easier or more, you will become more well versed with the topic. Okay, so a key morphological difference, as I have told you again and again, between apoptosis and necrosis is failure of apoptosis is contributing to what? Hallmark of biochemical feature of apoptosis is, and chromatin condensation during apoptosis occurs due to which of these factors? Something that I think it's very important that we have discussed this already, and it will help you answer these questions also. Yes, please use the chat box to answer. Okay, few more and then I will CABA. Okay. Read the question properly, okay? Morphological feature, not process. Okay, I'll give the answer, maybe 30 seconds more, just for people if they're attempting. Okay, I will give the answers. So BA and BNA. I'll tell why BNA, and I know why you have written C also. So the first question says a key morphological difference between apoptosis and necrosis. Morphological difference is basically something that you observe in the structure of the cell, right? So apoptosis shows membrane blebbing, right? So these protrusions without membrane rupture. Necrosis shows swelling and rupture with inflammation. So when you are observing under a microscope for morphological phenotype, what you will observe is a apoptosis cell will have these blebbing and these necrosis ones will don't not have. Inflammation is a process by which swelling takes place. So if you have to really write, you need to write that in the place of necrosis, there will be swelling. So because you're looking for morphological difference. That's why the answer here will be membrane blebbing. I hope that's clear and why the answer here will be morphological features. Okay. Like why for morphological feature, we are adding blebbing. Second one says failure. If you have any doubt, please do let me know. I will definitely try to clear it up. Okay. Failure of apoptosis can contribute to autoimmune disease, as I have discussed, and also, as I mentioned, cancer progression. Hallmark biochemical feature of apoptosis is DNA laddering. Laddering is basically so DNA is basically these long chain of your nucleotides together, right? Now, when you have DNA damage or when you have your apoptosis, basically fragments the DNA. It basically chops the DNA into smaller fragments, and that becomes a series of fragments back to back. So it looks like ladder. So it's DNA ladderization. That's what it means by DNA ladderization. Okay, and the last one says chromatin condensation during apoptosis occurs due to caspase activation that we have just discussed. As soon as your caspases become activated because of your respective pathways, they start cleaving nuclear proteins, and that leads to chromatin condensation. Right? Next one. So, initiator caspases primarily function to do what is when initiator caspase come, what do they do? Caspase 9 is which pathway? This is what I said. These are the kind of question that will come. Caspase 8, and then CARD and DED, which we will discuss. Where do these domains help in caspases? What are these used for? BBBC. Anyone else? Okay. 30 seconds more and then we can discuss the answers. Okay. I'll give the answers. Okay. So B, 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 and C, as we saw in the chat box. So so the first question is basically initiator caspases primarily function to activate execution of caspases. This is very easy to understand. As I told you, in both the pathway, the first thing to get activated is caspases, initiator caspases. Which one you need to remember? Which one is for 8 and which one is for 9? We will see that. And they both of them activate the executioner caspases. Okay. Next is caspase 9. As I told you, you should remember these is more important. So caspase 9 is from intrinsic pathway. Caspase 8 is from the death receptor pathway. Okay. And this card and dead are domains and caspases which are important for protein-protein interaction. So let me tell you the full form. The full form would have given away the function quickly. So card is basically caspase recruitment domain. So you can imagine that since these are interaction motifs, they are basically recruitment motifs. So they are interactive motifs and they basically are mediating the interactions between different domains which are basically working to activate the caspases, right? And the next one is death effector domain. DED is death effector. Effector means something which is functioning afterward. So death effector domain. And now you can imagine what it would be functioning on. So this domain is found in procaspases and what it does is it regulates the caspase activation. So we did this, right? We know procaspase is found in the extrinsic pathway, which you can see on this. If you remember from the slide, you have procaspase 8 and then caspase 8 and then caspase 3, right? So that's how that effector domain will come into play. Okay. And what was the underlying feature here? That it was involving protein-protein interaction. Okay. Next one, the PRO domain now. So you can already see this caspase protein is huge and it has so many domains, which is why it is able to interact with so many downstream effectors. So pro domain in caspase is important for which of these activity. ICAT is what is ICAT first of all? Now you have ROCK1 cleavage during apoptosis and intrinsic pathway. All these terminologies have been there in your notes. That's why to have them up on slide, you can use it up as your reference when you are trying to study it again. Anyone? Okay, I can see one person typing. Okay, all B. Fine. Anyone else? All right. I will just move ahead. Okay, so all B's in this set. So the first one is PRO domain in Caspace is important for recruitment and activation. So just for your understanding, PRO domain or the pro domain is basically the domain that contains the card and the debt. So that's why ultimately it basically leads to recruitment and activation. Okay. Okay. The next one is ICAD functions. ICAD is basically the ICAD full form is inhibitor of CAD. What is CAD? CAD is caspase activate DNAs. Okay, so caspase activated DNAs, anything that um, the enzyme DNAs which is activated by caspase. So this happens when your apoptosis has to take place. Okay, so that complex is called CAD, which functions under caspase activity. When there is no caspase, it has to be inhibited. So this ICAD basically inhibits those. Now, this ICAD has to be removed when your DNA fragmentation has to take place. So caspase 3, as soon as it gets activated, it first cleaves off this ICAD. As soon as the inhibition of the CAD goes away, the CAD gets activated. CAD basically means the DNase enzyme. DNase enzyme activated leads to fragmentation of DNA, and that leads to DNA laddering. So I hope you can imagine, you can make the framework of how this apoptosis pathways are happening. Okay. And next you have ROC1, which is ROC1 cleavage during apoptosis contributes to membrane blebbing. So this ROC1 is basically a protein serine threonine kinase associated with Rho. So it's basically a important, so the full form is Rho associated coil containing protein kinase. So you don't have to go into much detail. What you just need to remember is this protein, as soon as when it is cleaved because of your caspase activation, it leads to damages in the cytoskeletal structure, and it leads to the contraction of your membrane forming the blebbing. The blebbing that we have been discussing so far is because of this particular protein, right? And the last one is intrinsic pathway is triggered by, as we have already discussed it so far, internal damage, and the internal damage out of this option was DNA damage. Okay. All right. The next one is the intrinsic pathway is initiated, which is again very easy. Now, again, the anti-apoptotic BCL2 family. This is, you need to remember, anti-apoptotic and pro-apoptotic. So I thought I'll just put a question on that. And the last one is apoptosome, which is basically in your intrinsic pathway when your cytochrome C is released from the mitochondria. What structure or which components lead up to the formation of apoptosome? Anyone else wants to attempt? Okay, I'll give the answers here because it's fairly easy. So the extrinsic pathway is initiated through, intrinsic is by DNA damage, internal problems. Extrinsic is by outside, external cues. And external cues, I said that because they bind to the receptors there and they bring death for the cell. These are called death receptors. For example, FAS and TNF. Okay. Anti-apoptotic BCL2 family proteins function to, here read the answers and then you would have understood it is for stabilizing mitochondrial membrane. Anti-apoptosis means something that do not promote apoptosis. So stabilizing your mitochondrial membrane would make sense because it will prevent apoptosis. The rest of the options, promoting release, activating caspase, DNA damage, is all for these are all effects of, or these are all after effects of apoptosis, and these are anti-apoptotic proteins. So these are basically trying to stabilize the mitochondrial membrane. Similarly, if you have pro-apoptotic membrane, which are like bags, they are basically promoting DNA or mitochondrial permeabilization to form the holes that releases the cytochrome C. Okay, and the last one here, apoptosome, I have already shown that in the slide. It basically is B, cytochrome C, which is released from the mitochondria, APAF protein, and caspase 9, which is activated, which is the initiator caspase here in intrinsic pathway. Okay. Okay. So as the next set of questions, as you do more questions, I hope you can visualize these pathways and becomes more easy and becomes more intuitive as you go ahead. Right. This set here says DNA damage induced apoptosis involves the activation of which of these proteins? Caspase 3 primarily acts as a or an neurons tightly regulate apoptosis because we know something about neurons very distinctively, and a pro survival checkpoint in neurons often. So and why am I putting neurons here? Because they were discussed as a specific case in the lecture notes. Hmm. Anyone? Okay, 30 seconds and then we go ahead. A, B, B, B. Okay. Okay, I will give the answers so that move ahead. So DNA damage induced apoptosis P53. The others ones are basically uh, comes after your later steps of apoptosis. Caspase 3, as we know, is an execution of caspase after 8 from extrinsic pathway or 9 from intrinsic pathway. Neurons tightly regulate apoptosis because they are post-mitotic. We know that they arrest their cell cycles, so they are post-mitotic. So that's why they regulate the apoptosis, because they don't undergo mitosis anymore. So any more loss will be irreversible in neurons. They don't go mitosis after, like they never undergo mitosis. We read this, right? So what do? How do they check that they never have this loss? Their cells don't die. So what do? They have high BCL2 expressions, right? And high BCL2 means there is prevention of mitochondrial permeabilization, no holes or no mitochondrial damage, so the neurons will be protected. So neurons, because they don't proliferate or they divide anymore. So they have their own checkpoint to maintain the structure, right? Okay, the next set, not the last set, but one of the last few sets of this topic. Which process involves, so this is, so these are questions from one of the assignments, so you will see them repetitive, but just try to answer them. Basically tells you how what kind of questions can come. So which process involves programmed cell death essential for development? And homeostasis is very, very easy. What is the primary characteristic of cellular senescence, which is also something that we know already. I think this question is repeated also. Okay, the accumulation of which type of cellular damage is a hallmark of aging, and which protein is the key regulator in intrinsic pathway. This probably is, these two are probably something that we did not explicitly discuss as a question answer session. This we did already, so this should be easy. Easy questions, so shouldn't take much time. Okay, I will give the answer so that we move ahead. Apoptosis, we know already. Permanent cell cycle arrest, we know already. Accumulation of which type of cellular damage? Everything. Protein misfolding, proteostasis. Lipid peroxidation, we know about the cellular metabolism, then the nutrient sensing problems, so all of them. DNA mutation, we already know about DNA damage. And regulator of apoptosis and intrinsic is BCL2 because we are concerned with the molecules that are involved in regulating BCL2. So BCL2, we know that it basically prevents mitochondrial whole like permeabilization. And the last set of questions so that we move ahead to the last topic of today's session, which is meiosis. But before that, which of this is not a characteristic of necrosis? A gain of function is basically promoting an activity. Okay. So gain of function mutation in which type of gene can contribute to cancer development. Okay. Which of the following is not a characteristic of apoptosis? And what is the primary function of IAP? So this is what I was missed out. So IAP is basically inhibitors of apoptosis protein. What would they carry out? Easy questions, quickly doable questions. Anybody? Okay, someone has written BBCC. Anyone else wants to attempt? We can maybe have 10 seconds for anyone to answer. Okay, we can move ahead. Okay, so which of these is not which of these is not a characteristic of necrosis? Dependence on high ATP levels, because apoptosis requires ATP, which is during the process of apoptosome formation and all the pathways, right? Membrane integrity is also there in necrosis. Cell lysis is also there in necrosis. The only thing that is different between necrosis and apoptosis again is inflammation. Okay. A gain of function mutation in which type of gene can contribute to cancer. Cancer genes are called oncogenes. Proto-oncogenes are genes which get activated and then can lead to cancer. So gain of function means basically, so the function of the gene is to promote cancer. Gain of function means a mutation has happened that has promoted the function, that has let it to gain the function. So gain of function of a proto-oncogene is basically it has got the ability or the functionality of being an oncogene now, and it can now induce cancer. Which of these is not a characteristic feature of apoptosis? Because I just mentioned it, the induction of inflammatory response is for necrosis. Apoptosis does not have inflammation because these apoptosis marks cells for phagosome or phagocytosis. Basically, macrophages ingest them. And lastly, what is the primary function of IAP, which is also the inhibitors of inhibitor of apoptosis proteins? It is basically to inhibit the caspase activity and suppress apoptosis. Okay. All right. So that brings an end to the two topics of this week's lecture notes. Now we come to the last one, which is meiosis, which is very interesting. So you can briefly, first of all, you can see that meiosis has first of all, two phases, meiosis 1 and meiosis 2. And then meiosis 1 has on the slide right now, four phases: prophase, metaphase, anaphase, telophase, just like how it was in mitosis. But there is an additional name behind it, which is called the 1, which is this is part of meiosis 1. So prophase 1, metaphase 1, anaphase 1, telophase 1. And after telophase 1, you have two, like meiosis 2, which is again the prophase, metaphase, anaphase, telophase. We will see why it is showing one cell here and why it is showing two cells here. But broadly, it is divided into two different parts of cell division stages, okay, and types as well, right? Okay, so what is those type? What did I mention? Basically, if you talk about meiosis, remember I told about mitosis being equational division. Everything is equally divided, so the daughter cell and the parent cell remain with the same number of chromosomes, and we did this entire activity of seeing what is chromosome number, how they are divided across the cells in mitosis. In meiosis, the idea is it's not so generally when you say cell cycle and when you come to the M phase, it's called mitotic cycle or mitotic phase. That's because it is the general mode of cell division. But meiosis is a specialized cell division because it happens only in germ cells. Okay, diploid to haploid, and the haploid germ cells that they basically lead to the formation of zygote when they fuse with another haploid germ cell. So a haploid germ cell from a father and a haploid germ cell from a mother fuse to form zygote, and the haploid and haploid content together give rise to the diploid content of the zygote. So this is a specialized cell division occurs in germ cells, and that's why when you say M phase, you mostly talk about mitosis, but meiosis is also very important. Now, meiosis has two divisions that I just mentioned, meiosis 1 and meiosis 2. Meiosis 1 is what makes it different from mitosis. Meiosis 2 is basically same as mitosis. Okay, meiosis 1 is called the reductional division. Meiosis 1 is where you have the division or the separate or the reduction in the chromosome number from being a diploid to a haploid. And then once you have these haploid cells, they basically undergo mitotic divisions, which is called the meiosis 2. Okay, and that's why you have these cells here, which I can just take you once again. Meiosis 1 basically starts with that single cell giving rise to two daughter cells. And now each of these daughter cells will undergo mitosis to give two more daughter cells, which ultimately leads to four cells. So one cell leads to four cells in meiosis, right? Okay. And remember the haploids, chromosome number one, it starts with a diploid, ends with an haploid. Okay. Now, as I mentioned briefly, you have prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 is basically the chromosome condensation and the nuclear envelope breakdowns crossing over. This crossing over should tell you that this is where the variation comes in natural or human beings or the zygotes, because there's something called homologous recombination that takes place between the chromosomes, and that leads to these different kind of alleles that we see, variations that we see, right? So prophase 1 is not that simple as it sounds here. Prophase 1 further consists of 5 more phases, which is called leptotin, zygotin, packaging, diplotin, and dikinesis. Let's try to understand. It seems very heavy, but it is, once you see through it, it becomes very easy to understand, right? Okay. So, right. So your prophase 1 is basically, as I just told you, it has more steps. So let's start with the first one, leptotin. Okay. Now, I hope you can visualize this. Let me just first tell you what happens and then we can see. So your chromosome begins to condense. This is exactly what happens in the prophase of mitosis also, right? Each chromosome here consists of 2 sister chromatids that we already established in the previous session. If you are still confused about chromatids, chromosomes, please refer to the previous problem solving session. That will help clear those concepts up. Okay. So each chromosomes consist of 2 sister chromatids, leptotin. Cohesin, we remember these names, right? Cohesin proteins are holding these chromatids together, right? Now, the first thing, remember, in this phase, you have also to be done homologous recombination. So for a recombination to take place, it's basically, let me try to see if I can show you. Okay, here you can see, right? There is a, so the red is from one chromosome and the blue is from one chromosome. And you have, otherwise you have this red upper arm, lower arm, 2 sister chromatids, right? Blue upper arm, lower arm for both sister chromatids. Now suddenly you go here, you suddenly have this blue arm with one red tail, red arm with one blue tail. This is happening because the content are getting recombined. So the DNA from here is shifting to here and the DNA from here is shifting to here. That's because of recombination. For this to happen, there has to be a breakage in the cell, so DNA, so that the DNA can nick off and then join with the opposite partners, right? That break is initiated in the first phase of prophase 1, leptotin. It's called double-stranded breaks, which is done by SPO1 or SPO11. Okay, so in your leptotin, you can see here the nicking will start. Okay, your condensation, because they will form a chromosome, and the nicking will start of these or double-stranded breaks for that matter will begin. Okay, initiate. Next phase is zygotin. Okay, in this, your homologous chromosome began pairing. Okay, so your homologous chromosomes, which are otherwise separated in the cell nucleus, will start coming closer to each other. So this is what is homologous chromosome? One from father, one from mother. So they come closer to each other. Okay. And then what do they form? They form something called synaptonemal complex. Just hold with me. Let's try to listen. I hope that it becomes clearer. I will just once again later summarize it for you. So formation of synaptonemal complex is basically you have this lateral element, central element, and your aligns, there is the homologous chromosomes are basically aligning with each other. And you see these markings here at the end, which is called the synaptonemal complex forming. That's exactly where they are starting to pair up. The next one is packeting. So in the first phase, breaks have started. They have started forming chromosome. In the next phase, the chromosomes become homologous pairs. Third phase, your synapses basically coming together is completed and your crossing over takes place. For this, you have the homologous recombination mediated by RAD51 DMC. Remember those DSBs, they just cross over each other and bind to the opposite partners. And that's how your genetic diversity is created, because the material for one of the set of parental chromosome moves to the other parental chromosome, right? Right. So your homologous recombination has taken place here in packeting, as you can see this marking here, right? Now you have diplotin, which is this chromosomes need to separate so that they can move ahead. So this breakage of the complex takes place and these homologs again become separating. And you have this chiasmata, no, this phase with this region which is undergone chromosome homologous recombination is called chiasmata, and you can see the chiasmata very clearly. And the last step is dikinesis, when your chromosomes have gone back to fully condensation and basically the homologous chromosomes have separated now, so they are moving separately. Like they have, so the synaptonemal complex is broken. So only at this chiasmata they are connected. Everything else is separated, and your nuclear membrane has started disintegrating. Spindle formation has taken place, which is something that we have already discussed. This last and the first step, leptotin and the diakinesis, is the end and the start of prophase. Everything else in here is for the recombination to take place. Okay. Right. And then you have metaphase, which does the same thing as it does in any other phases that we have studied. So we come back to metaphase now. Now, in metaphase one, your homologous chromosomes, which are the ones with the, pairing, they will come and align. And random orientation leads to independent assortment. Means what? It basically, so the cell move like this, right? All these four will move this side, all these six will move this side, all these six will move this side. But the orientation of the homologous chromosome is not governed. Like for example, in one, let's call this as pole one, pole two. So this pair is the red one is facing up and the blue one is facing down. In the other one also red is facing up, blue and facing down. But in the third one, your blue is facing up and red is facing down. What happens here? When your this cell will form, it will have chromosomes from, let's say, red is parent, father. So father, father, mother. And this one will have father, mother, mother, father. So you have independently assortment of your DNA or your chromosome. This is also one of the tenets that we had read about genetics when we know about the independent assortments of genes, right? Or alleles for that matter here, right? So your metaphase, they have all lined up in random orientation, and in anaphase, the homolog chromosome separate. The first thing to get separated here is the chromosomes. It is in contrast to mitosis because there your sister chromatids separate. Right now, your sister chromatids are going uh, together to the opposite pole. So what has separated chromosome 1, chromosome 1, chromosome 2, chromosome 2. But the chromosome 1 sister chromatids haven't separated yet, right? Right? And at the end, you have telophase and cytokinesis. The same way you have two cells formation and the cell breakdown. Now, in the next phase is when your mitosis will take place because now again you have these sister chromatids and this sister chromatids will separate and go to the opposite poles. So that's exactly what happens here. When did this division happen? See, you started with 2n, but because your 2n, which is contributed by father and mother, those chromosomes itself got separated. Now individual cells have only half the genomic content. That's when your division has, like reduction and division has taken place. Here, in both the cell, whatever content is present from, so two copies of mother, two copies of father for this chromosome, they will be separated, and the cells ultimately at the end, because the sister chromatids are separate, the content will still be the same. So that's why in your meiosis 2, prophase 2, it basically chromosome condensed, become more condensed, spindle apparatus form, metaphase 2, they align, anaphase 2, they break up, the separation of sister chromatids take place, they move to the opposite poles, and four haploid daughter cells, two from each new daughter cell are formed. Okay, I hope this is all easy. Let's try to answer questions so that they become easier. Okay, let's catch up with that. Okay, let's try to discuss more. We'll definitely understand it better. So the primary function of meiosis is to do what? Meiosis 1 differs from meiosis 2 how? Uh, Crossing over occurs. So these are the things that you should remember. At which phase, at which step, what is taking over, what's going on. And the synaptonemal complex forms between which of these pairs? Yeah. Not very difficult. Try to think on the answers. This will help you understand the complicated 8 steps of meiosis better. Anyone wants to attempt? Okay, let's give the answers, which are easy for that matter now here. All right. So C, B, A, and B. So primary purpose of meiosis is to basically generate haploid gametes, right? It happens only in germ cells. Meiosis 1 differs from meiosis 2 because meiosis 1 separates homologous chromosomes. Remember this distinction, very important. Sister chromatid separation happens in mitosis or meiosis 2. Okay, right. Crossing over takes place in prophase 1. Which step of prophase 1? Sorry, here. Okay, in packaging. And synaptonemal complex forms between homologous chromosome that we have already established. Next, the significant outcome of meiosis. Significant outcome of meiosis. So, what is the outcome of meiosis? What is the role of crossing over? Why do you at all need this crossing over? Which stage of meiosis is similar to mitosis? These are the kind of words that I have used also. And these are the kind of questions that will come also. So that's why it's easy. Which of the following event occurs only in meiosis and not in mitosis? The more you answer, this becomes very easy actually. Anyone wants to attempt? Okay. So let me give the answers. Yeah. D, B, C, and C. Okay. Significant outcome of meiosis is increasing genetic variation because of homologous recombination, reduction of chromosome number by half, formation of gametes. All of these are correct. What is the role of crossing over during meiosis? It increases genetic diversity because of the recombination. That's why you need this crossing over. It's also called homologous recombination, as we have established. Which stage of meiosis is most similar to mitosis? Anaphase 2, because that's when your sister chromatids separate in mitosis, even in meiosis, right? And the last one here is, which of the following event mostly occurs only in meiosis and not in mitosis? Pairing of homologous chromosome, because separation of sister chromatids happens in both. DNA replication is needed for both to form the second pair of sister chromatid. Chromosome condensation happens again in both the mitosis and meiosis, right? Okay, next one. Which type of chromatin is transcriptionally active? Non-disjunction during meiosis. Non-disjunction is basically the breaking or separating during meiosis can result in which disorder? Which structure holds homologous chromosomes together during chromosomes? We remember we told that, so this is discussed already, which one holds them together. And independent assortment, which is also I have discussed right now, in meiosis happens due to which reason? Okay, we will do this quickly. I will wait on each slide for a minute for you to answer because we have questions to complete, and then we can, like, the questions can be discussed. Okay, so the answers, as someone has mentioned, A, D, C, and B. Okay, which one is transcriptionally active? It's euchromatin. These are basically depending on which packing of your histones and your DNA. So which one is transcriptionally active is less compactly packed. Okay, because it has to be accessible to the other proteins. Non-disjunction is basically separation of the chromosomes, and that if doesn't happen, it leads to Down syndrome. Down syndrome is also called trisomy of 21st chromosome. Basically, in your 21 chromosome has three chromosomes together instead of two. Okay, so that's because your chromosome does not separate properly. Disjunction is basically separation. Okay, and as we know, homologous chromosomes are held together by synaptonemal complex. And independent assortment, as I've explained, it is because of random orientation of homologous chromosome at metaphase. Next one, why do you need the synaptonemal complex? Homologous recombination increases what? Double-stranded breaks in meiosis are initiated by which of these proteins? And RAD51, which I did mention, is important for what? I will wait for just 1 minute and we will give the answers. 30 more seconds. So if you feel that this is too fast, and because I want to finish and show you all the questions, the videos will be there. So you can pause the video and think of the answers and then try to see if the answers match with the correct answer that I display on the screen. Okay. All right. So the answers for this are the synaptonemal complex is essential for homolog pairing, as will know already. And they are basically stabilizing the homolog pairing and enabling crossing over. Homologous recombination increase variation because I told you the genetic material is exchanged between the parental chromosomes. Double strand breaks in meiosis, which basically is required for the recombination to take place, is done by SPO11. RAT51, I had mentioned, it is involved in the recombination. So there is something called strand invasion. So RAT51 does strand invasion, and the name should give you an idea of what's happening. So you are invading the other strand. So you have these two strands which are from different parents, and the breakage is this side. So this should basically come and bind to this. It will go and invade this strand, and this one has to come bottom. So it will form this X-like structure. So it's called strand invasion. You're invading the other strand. Okay, so that's done by RAD51. Okay, next one. During leptotin stage, what happens? Synapses is, so this is important. This basically tells you about the different stages and it also helps you revise quickly. Synapses is completed in which stage? Chiasmata is visible in which stage? When do we actually see the chiasmata forming? And separation of the homologous chromosomes occurs during which phase? Okay, a minute on this and then we move ahead. Okay, 30 seconds. ABCA. Okay, I will show the answer. Yes, it's ABCA. So let's do it. During leptotene is basically the first step, the first stage in your meiosis, prophase one, right? So your leptotene has chromosome condensation. Synapses is basically happening in Pac18. Okay, in Pac18 you have the synaptocene complex formation completion. Okay, before that you have zygote where it starts to form. Okay, chiasmata becomes visible in diplotin when your crossover has taken place and they have started to move ahead, right? So when they have begun to separating, and separation of homologous chromosomes take place in anaphase 1. Okay, all right. Next set, sorry, yeah. Separation of sister chromatids occurs in which stage? Cohescent proteins are cleaved during which stage? The reduction of chromosome number occurs in which stage? And non-destruction results in which of these problems? 1 minute again, and then we will look at the answers. Sorry about that. Yeah. Okay, so let me give you the answers since we have only few time remaining. Separation of sister chromatids occur in meiosis 2, right? Cohesion proteins are cleaved in both the stages of NFS 1 and NFS 2 because they have to move ahead. Reduction of chromosome occurs in meiosis 1, this we have established already, and not what happens in direct key protein maintaining sister chromatic cohesion is which one? Okay, so I will give the answers here. Just a second. Right. Okay. Coming to the answers. Central element of synaptonemal complex contains transverse filaments. So these basically connect. So as we saw in synaptonemal complex also, right, there was this kind of strands between the chromosomes. So that's transverse filaments. Homologous recombination pathway resembles the mitotic DNA repair. So basically, it's just for information that the strand invasion that we said, I said that there is strand invasion that is similar to DNA repair mechanisms. Okay. Okay. During diakinesis, which is the last step of your prophase one, you have nuclear envelope breakdown because I told you, remember the last and the first step of your prophase one matches to the actual or matches to the prophase of your mitosis phase, right? So your diakinesis is similar to your prophase preferred is for metaphase 1, right? So it is similar to the mitosis. And the next one is a key sorry, just a second. Yeah, a key protein maintaining sister chromatic cohesions at centromere, as we all know, it is your sugosine. Okay, so you should remember that when a sister chromatin cohesion, its centromeres is because of Sugosian in meiosis 1, because they need to be kept together, they cannot go separated, right? Okay, and the last questions from previous assignments. So which stage in meiosis is characterized by the occurrence of crossing over between homologous chromosomes, and non-disjunction of chromosome occur due to which reason, which already we have discussed so far. So it should be easy. Okay, someone, okay, I see someone typing. I'll just wait. D and D. All right, that's correct. So, which stage in meiosis characterized by the occurrence of crossing over between chromosomes is packetin. So you will remember leptotin, zygotin, and then you have packetin, and then diplotin and dikinesis. Okay, so packetin. And disjunction, which is failure to separation of the chromosomes, that's because what is the possible reason why can't they separate? They can't separate because probably there is premature segregation of sister chromatids. So your one of the sister chromatids went ahead and separated instead of the chromosomes, or your pairing did not take properly, so they are not aligned properly, or there is missegregation of chromosomes. Basically, that basically means that they should have moved this way, but so one of the pairs should have gone to the opposite pole. Both the pairs move to the opposite pole, to the same pole. That's when your missegregation takes place. So all of this is basically referred to as non-disjunction of chromosome, because disjunction means that both the chromosome pairs are in different pole. When that does not happen, that's referred to as non-disjunction, and that can happen because of all of these reasons. And the last one for today, the correct order of prophase one, because we have been so far discussing on these different stages. So what is the one final, like this is the final slide to tell you which one follows after the other. And the chromosomal number is reduced to half during which of the stage of meiosis, 1 or 2, which is very easy. Yeah, seeing many people are typing. That's exactly because it's very easy. So in general, like for these kind of multiple steps questions, you can make an acronym for yourself, some shortcut to remember them so that you don't forget them. Yeah. Okay. Everyone has given the answers A and B. Okay. Anyone else wants to attempt? Okay. I'll let you. It's already 8 o'clock. Okay. First one is leptotin, zygotin, packetin, diplotin, dikinesis. So you can make a short of acronym of it for yourself if you want to remember it like this. But yeah, this is the correct order. And the half, which takes place in the meiosis 1. Once again, meiosis 2 is similar to mitosis. Mitosis is an equational division, so meiosis 2 is also equational. Reductional is the first one of meiosis 1, right? Okay, that brings an end to today's session. And if you have any doubts, you can post it in the chat box. This recording will be available to you. This presentation will be available to you. And yeah, if we have no doubt, we can see each other in the next class, which is next Monday, same time. Thank you so much for attending. Thank you. Thank you very much.